Welcome to MOOC's online course on cancer drug discovery and development. Myself, Dr. K. Kumar Reddy. In this module, we are going to discuss on ligand-based drug design, different methods available in ligand-based drug design that we can use in drug discovery workflow and virtual screening. If you remember from the previous lecture, computer-aided drug design is an approach which uses the knowledge of target and ligand molecule in the process of drug discovery workflow. Based on the knowledge being used by these computer-aided drug design methods, these methods can be classified into two types: structure-based drug discovery and ligand-based drug discovery. We have discussed about structure-based drug discovery in the previous module. Now, in this module, we are going to briefly discuss on ligand-based drug discovery. Ligand-based drug discovery is a method where we use the knowledge of known active compounds that bind to the target of our interest in the drug discovery workflow. Ligand-based drug discovery is used when there is no prior knowledge of the receptor structure. To use ligand-based drug discovery, at least we need one active compound that is known to bind to the target of our interest. Ligand-based drug design is an approach used in the absence of the receptor 3D structural information, and it relies on the knowledge of the molecule that bind to the biological target of interest. These methods use a set of reference structures collected from different studies that are known to interact with the target of interest, analyze their 2D and 3D structure. The overall goal of these methods is to represent these compounds in such a way that physicochemical properties most important for their desired interactions are retained, whereas extraneous information not relevant to the interaction is discarded. Ligand-based drug discovery is based on similar property principle, which states that molecules with structurally similar are likely to have similar properties. Two fundamental approaches of ligand-based drug discovery are selection of compounds based on chemical similarity of known actives by using some similarity measure. The second approach is construction of a model that can predict the binding activity of unknown molecules from their chemical structure. Ligand-based drug design methods use different methods for describing the features of small molecules using computational algorithms that balance efficiency and information content. Molecular descriptors or features can be structural as well as physicochemical, and these descriptors are generated by using knowledge-based molecular mechanics or quantum mechanics based methods, and molecular descriptors can be classified based on their dimensionality as one-dimensional, two-dimensional, and three-dimensional. The examples of molecular descriptors include molecular weight, volume, surface area, rotatable bonds, electronegatives, symmetry, functional composition, solvation properties, and many others. So, coming to methods that are available in ligand-based drug discovery, there are three different methods in ligand-based drug discovery. The first method is molecular fingerprint and similarity search, and the second method is quantitative structure activity relationship, and third method is pharmacophore modeling. Coming to molecular fingerprint and similarity search method, these molecular fingerprint based techniques attempt to represent molecules in such a way as to allow rapid structural comparison in an effort to identify structurally similar molecules or to cluster collections based on structural similarity. These methods are less hypothesis driven and less computationally expensive than Pharmacophore and QSAR based methods. They rely entirely on the chemical structure and omit the biological activity of the compound, making this approach more qualitative. Additionally, fingerprint-based methods consider all parts of the molecule equally and avoid focusing on only part of a molecule that are thought to be most important for its biological activity. Unlike similarity search-based methods, QSAR and pharmacophore modeling methods consider physicochemical properties most important for their desired interaction, and by discarding extraneous information not relevant to the desired interactions. So, the second ligand-based drug discovery approach is pharmacophore modeling. Pharmacophore modeling is based on the principle that if a group of ligands are known to inhibit a particular biological target, this means all these compounds that are inhibiting the biological target should possess some common physicochemical properties that may be responsible for the observed biological activity. The typical types of interactions recognized by pharmacophore modeling include hydrogen bond acceptor, hydrogen bond donor, ring aromatic feature, hydrophobic interaction, positive and negative ionizable properties. So pharmacophore mapping or modeling involves basically three processes: finding the features required for particular biological activity, determining the molecular conformation required, developing superposition or alignment rule for this series of compounds. All these methods use activity in the development of Pharmacophore model. We can generate two different types of Pharmacophore model. One is qualitative Pharmacophore model, other one is quantitative Pharmacophore model. Generating qualitative Pharmacophore model have the active ligands. In this method, what we do is we take the active ligands, try to identify what are the common physicochemical properties that are present in the group of ligands that we have taken. So it basically gives what are the physicochemical properties that are necessary for any compound to interact with the target of our interest. The second method is quantitative Pharmacophore model. For generating quantitative Pharmacophore model, we need to have a set of ligands that are known to inhibit the target of our interest. The ligands, the set should be structurally very diverse and should cover a wide range of activities. So let us consider if you have a compound with a specific structural scaffold and known to have a biological activity against a target. If you don't include this particular compound in your Pharmacophore model generation, you will miss the compounds in the database that have similar structural fold. For successful Pharmacophore model generation, we need to have a good set of starting structures. The overview of how Pharmacophore based model is generated can be seen in the figure. So first you have a set of molecules from which we can generate a Pharmacophore model. Then we use this Pharmacophore model to screen a database of compounds, identify hit compounds. The identified hit compounds again were then biologically evaluated to identify lead compound. If any of the hit compound is biologically active, you can use the newly identified in the initial set of compounds again you can generate a new Pharmacophore model. This cycle we can perform until we get a compound which has good biological activity against your target. A typical Pharmacophore can be seen in the figure. This Pharmacophore has been generated against human 5-lipoxygenases. For this study, we have taken a set of 89 compounds that are known to inhibit human 5-lipoxygenase from 18 different studies. The activities are ranged from 0.003 micromolar to 41 micromolar. We have separated the 89 compounds into two different sets. The first set is training set and the second one is test set. Training set is a set which we use for the generation of the Pharmacophore model. Once you have generated the Pharmacophore model by training set, we validate this model by its ability to accurately predict the binding affinity of the test set compound. Once this Pharmacophore is ready, we can use this Pharmacophore for screening a chemical database. In this study, we have taken the Pharmacophore model and we have used this model to screen against Maybridge database to identify from the virtual screening. We have taken the first 2000 compounds, further analyzed them using docking and visualization studies and selected 15 compounds, out of which we have ordered 5 compounds and evaluated their ability to inhibit human 5-lipoxygenase. Out of the 5 compounds tested, 2 compounds showed inhibition against human 5-lipoxygenase with an IC50 values of 14 and 35 micromolar. Another most important thing we need to remember in Pharmacophore model is not only about the chemical features but also the orientation of these chemical features. Pharmacophore model we have generated against human 5-lipoxygenase. We have identified 4 physicochemical properties that are necessary for inhibiting the human 5-lipoxygenase. The 4 physicochemical properties are 2 hydrogen bond acceptor which are represented in green color, 1 ring aromatic feature which is represented in red, and 1 hydrophobic feature which is represented in cyan. Coming to third method of ligand-based drug discovery, which is quantitative structure-activity relationship. As the name suggests, this method correlates molecular structures with biological properties. The QSAR methods are based on assumption that the structure of a molecule must contain the features that are responsible for its physical, chemical or biological activities. This method is defined as a process that quantitatively correlates structural or molecular properties, which we call as descriptors, with functions like physiochemical, properties, biological activities and toxicity, etc. The general workflow of QSAR can be seen in the figure. In this QSAR approach include a collection of active and inactive compounds and generate molecular descriptors that describe the physicochemical or structural properties of the compound, and then generate a model that identifies the relationship between the descriptors and their experimental activity. There are different types of descriptors and there are different methods for doing model. Now let us discuss how we can use QSAR in our drug discovery workflow by taking an example. In one of our study where we used 3D QSAR method in the lead optimization of 5-hydroxyindole carboxylate derivatives that inhibit 5-lipoxygenase. For this study, we have taken COMFA, COMSA, which are 3D descriptors, and we use PLS method for modeling the QSAR. A total of 40 compounds were taken for this study, out of which 30 were taken as a training set to generate the model and 10 were used as a test set to evaluate the model. We have performed both COMFA and COMSA analysis for lead optimization in this study. COMFA, which means compared to field molecular analysis, is a 3D QSAR technique that aligns molecules and extracts aligned features that can be related to biological activity. This method focuses on the alignment of molecular interaction field rather than the features of each individual atom. In this method, molecules are aligned based on their 3D structures on a grid, and the values of steric, which are a van der Waals interaction, and electrostatic potential energies, which are nothing but Coulombic interactions, are calculated at each grid point. COM is the extension of COMFA, considers hydrogen bonding and hydrophobic terms in addition to steric and Coulombic interaction. The COMFA and COMSA models we have generated for 5-hydroxyindole 3-carboxylate derivatives that inhibit 5-lipoxygenase can be seen in the figure. Each counter is specific for one functional group and is represented in two colors. One region represents where that modification of that particular functional group can increase or decrease the activity. For example, if you see the first counter, it is for steric groups, which are nothing but van der Waals interaction. If you add steric groups in the yellow region, it decreases the activity of the modified compound against 5-lipoxygenase. Whereas if you add the steric groups at the green region, that will increase the inhibitory activity of the modified compound against 5-lipoxygenase. The second one is for electrostatic, where blue region shows where electronegative groups decrease the activity, whereas red region show where electronegativity group increase the activity. Similarly, in COMSA also, there are different colors, different ways of representing the functional groups, and based on this COMFA-COMSA data we have generated, we can modify the compounds in such a way that it have higher binding affinity against your target protein. Now we know what are the different types of computer-aided drug design methods that can be used in drug discovery workflow. The most widely used computer-aided drug discovery approach in drug discovery workflow is virtual screening. Virtual screening is an in silico technique that is used to search large compound database to identify potential lead compound. In this method, we use either structure-based or ligand-based drug design approach to score, rank, filter a set of chemical structures from a chemical database and select them for biological testing. Based on the approach we are using in the virtual screening, these methods can be broadly classified as structure-based virtual screening and ligand-based virtual screening. In general, virtual screening uses computational methods for screening chemical libraries for identification of lead compound against intended drug target. It is a step-by-step method with a sequential filter that will narrow down and choose lead-like compounds by scoring, ranking, and filtering molecules that are likely to bind to a target. There are various chemical databases that are available online. Some of them are in the public domain and some are commercially available. As you can see in the table, these are the different chemical databases that we can use for identification of fit compounds against your target protein. The most commonly used filter in the virtual screening is Lipinski rule of 5. Lipinski rule of 5 defines the druggability of a novel chemical compound. So rules include log p value less than or equal to 5, molecular weight less than or equal to 500 Daltons, number of hydrogen bond acceptors less than or equal to 10, and number of hydrogen bond donors less than or equal to 5. If a compound follow the above mentioned rule, that the compound has for doing structure-based virtual screening, we can use either we can use docking or receptor-based pharmacophore modeling in the virtual. In this study, the authors used molecular docking approach for virtual screening to identify novel EGFR tyrosine kinase inhibitors. They have taken the crystal structure of EGFR kinase and they have screened SPECS database that contains around 2 lakh compounds and identified 43 hit compounds, out of which 13 compounds showed inhibition against EGFR tyrosine kinase. Coming to the next virtual screening approach, which is ligand-based virtual screening. For performing ligand-based virtual screening, we need at least one ligand that can bind to the target of our interest. If you have set of ligands, we can generate Pharmacophore model for the ligand, and you can use this model to screen chemical databases to identify hit compounds. For example, in this study, authors have taken 5 natural compounds that bind to androgen receptor downregulating agents and generated a Pharmacophore model. This Pharmacophore model was then used to screen against Maybridge database that contained approximately 60,000 of compounds and NCI database that contained more than 2 lakh compounds to identify potential hit compound. This search resulted in identification of 5 novel compounds that showed inhibition values in micromolar range. I hope by the end of this lecture you got a basic idea of what ligand-based drug discovery approaches are and how you can use them in drug discovery workflow. In addition, I also hope that you got a basic idea of what is virtual screening, how we can use virtual screening in drug discovery workflow. Thank you.